everyone. We will see about the other poikilocytes. The next set will include target cell, ovalocytes, acanthocytes, and stomatocytes. Target cells, these are formed because of excess membrane compared to the amount of hemoglobin inside. This is exactly the opposite of spherocytosis, where you get decrease in surface area compared to the hemoglobin volume. Whereas here, there is an increase in the surface area relative to the volume. In the sense, there is an excess membrane compared to the amount of hemoglobin inside the RBC. So, the membranes just becomes very redundant and you get ups and downs. When the ups are there, here the hemoglobin is more, so you get a colored ring like thing, and then here the membranes are approximating, and then in the center again there is a concave and convex membrane. In between them there is hemoglobin, resulting in the target appearance. So target cells are seen when there is increased membrane. This is usually seen in cases of hemoglobinopathies where there is in decrease in hemoglobin synthesis as the pathogenesis. Also in liver diseases where there is excess membrane. So liver diseases and in your hemoglobinopathies you get the target cells. These are also known as podocytes. The next will be an ovalocyte. So ovalocytes is a genetic defect where there is a defect in the cytoskeletal protein. So what happens is normal RBCs can actually, when they are squeezing through these sinusoids and all, they just there's this shape again they become oval as they just go along, and then again they regain their spherical shape or the disc shape. But here, because of the cytoskeletal protein abnormality, what happens is the membrane sliding over the cytoskeletal protein. You know that the RBC has a lipid membrane and then you have the cytoskeletal protein. So when there is a defect, this regaining of the shape when they just squeeze through these will not happen, and so they will retain the acquired oval shape, and this results in many RBCs becoming ovalocytes. Acanthocytes, because of the balance loss between cholesterol and phospholipids. What happens is usually you will have an outer lipid and inner lipid membrane in an RBC, as we all know. So the outer layer, lipid layer, will have more of cholesterol now compared to the inner layer. So this results in speculated projections. There is an imbalance between the outer layer and inner layer, inner lipid layer cholesterol content. So this results in spikes of the membrane because the outer membrane becomes very rubbery because of the high cholesterol. So the inner membrane has less cholesterol in it. So this results in this spikes coming out, and they are seen in liver diseases. So the last one will be your stomatocytes, where you get a problem with the membrane permeability. So because of the lipid distribution changes, there is membrane permeability change, and that results in a morphology like your slit like areas, like a mouth like area in the RBC. We will see more detail of this because this is just a brief about the pathogenesis. We will see individually with animated videos so that you understand them better. Thank you. We will see the next bite cell, blister cells, and the RBC inclusions in our coming part 3. And as I always say, please like, subscribe, and share if you like our videos. Thank you.